So in this section, we're going to actually start talking about things that are alive finally in here. Biology, something that's alive, yay. So we're looking at the cells, the overview of the cells. Now it is a cell biology class, so obviously the rest of the stuff is looking at things that are going on in cells in this class, but we're getting started with the living stuff finally. So cells are too small to study with the naked eye. The only cell that's a human cell that you might be able to see with the naked eye, and just barely, is the egg. That is the largest human cell in there. It's, if your eyes aren't really good, you're not going to see it with the naked eye, and if it's not stained, you're not going to be able to see it with the naked eye really in there. It would be a case of maybe just barely being able to see it in a petri dish, and anybody who's going to work with them is going to have a microscope to assist them with that. The light microscope is the most common one, and with this one, visible light is going to pass through the specimen and then through a glass lens, and the light will refract to magnify the specimen up to about 1,000 times. That's about the most you can get with a light microscope, is 1,000 magnification. And when we talk about magnification, it's the ratio of the object's size to its real size. So that's where that 1,000 times, it appears 1,000 times bigger to your eye when you're looking through the microscope. The resolution is going to be the clarity of the image, and the way we define that is the minimum distance 2 points can be separated and still distinguished as 2 points. So at some point, if things are too close together, when you look at it, it's going to look like 1 point. You will look at it and you'll go, I can't tell, is that 1 or is that 2 there? So that's what we talk about with resolution in there. For the light microscope, it's about 0.2 microns or 200 nanometers. And that distance is going to be limited by the shortest wavelength of the light used, because the wavelengths of the light being refracted are what's doing the magnification in there. The contrast is going to accentuate the difference in the parts of the sample, so making some of it look dark, some of it look light in there, and that's why we stain it, to help increase that. When you look at a wet mount in there, when you looked at the stuff from the pond water that wasn't stained, you'll notice it was harder to see. It just kind of looked like maybe there was something kind of sticky in there, like a clear glue in there maybe, is what it appeared like to you. And the stain will help to be able to see the details in something like that. So cell walls were first seen by Robert Hooke in 1665. And Anton van Leeuwenhoek was a lens maker that made the first microscope. The first microscope was essentially a magnifying glass in there. He was a lens maker, and he actually called what he saw beasties in there. So that was how they saw them. It wasn't an elaborate microscope like you're picturing yourself using. It was magnifying glass in there that they used. Most organelles, which are the membrane compartments in the cells, are going to be too small to see with the light microscope. So when you've looked at cells, you notice you didn't see a whole lot in the middle. You get to see kind of the blob shape and a dark area in the middle and not a whole lot else. That's pretty typical with a light microscope. When an electron microscope is used, these were introduced in the 1950s. They're going to focus beams of electrons through the specimen or onto the surface. And their theoretical resolution is 0.0002 nanometers based on the wavelengths of those electron beams in there. That's in theory that it could get that well, but in practice we're still only able to get it back down to about 2 nanometers, which is still a lot more detail than what you'll see with the light microscope. It's going to reveal more, but it takes a lot more preparation to make these. It's going to kill the cells. They have to be prepared in a vacuum in there so that there's no light in there to distort things. They also use really toxic heavy metal dyes on electron microscopes. And the electron microscopes are extremely expensive. So odds are, unless something changes in the near future, none of us are ever going to handle an electron microscope in there. Up at the UW, they never even, I know they had one, but they never allowed us to, we didn't even know where it was, kind of thing in there. They're extremely expensive pieces of equipment that they're just not going to let people at. The ultrastructure is going to be the cellular anatomy that's revealed by the electron microscope. There's two variations in there. You've got the scanning electron microscopes that are going to give the image of the object. So you've probably seen those where they look like little planets with these rough surfaces on there. And then you've got the transmission electron microscopy that's going to study the internal structures, and those have to be stained with the heavy metals as well. Most electron microscope images are going to be black and white, but they can be color enhanced with a computer in there. So cytology is just the study of cells. The ology ending means the study of in there, and cyto is cell. So cell fractionation is going to take apart cells and separate the major organelles and subcellular structures with a centrifuge. The reason you would do this is if you wanted to study some of those cellular components in bulk, that way you could separate it out and get a bunch of them to study. Otherwise, it's not very useful in terms of studying a living cell because you're going to kill it when you take all the organelles out of it. So with the structures of cells, we divide those into the two main categories, prokaryotic cells and eukaryotic cells in there. Eukaryotic means true cell. And what's going to separate them is the internal membrane compartments. Eukaryotic cells have internal membrane compartments. The prokaryotes don't. So the plasma membrane that both are going to have is going to be a selective barrier around the outside of cells. That is its wall between the cell and the outside world. Some of them will have additional walls or capsules around there, but your animal cells are going to just have that membrane that separates them. Some common organelles, all cells are going to have chromosomes that carry your genes. All cells are going to have ribosomes. There's plenty of other organelles that we will talk about with specific cells. The DNA in the prokaryotic cells and eukaryotic cells is a little bit different. In the eukaryotic cells, it's going to be in the nucleus, and it's going to be surrounded by a double membrane. In your prokaryotic cells, they don't have a nucleus, so they cluster their DNA in one specific region, and we call that the nucleoid region. Your cytoplasm is going to be the interior of the cell. It's a cytoplasm in both prokaryotic cells and eukaryotic cells. The interior of the prokaryotic cells is going to be the cell area between the plasma membrane, or it's all the area in the prokaryotic cell is between the plasma membrane and the nucleus in the eukaryotic cell. So I guess it's not worded as confusing as I thought it was. The size, eukaryotic cells are generally much larger than prokaryotic cells. Red blood cells are pretty small. They're on the smaller end of eukaryotic cells. So that's why you have to look at them with oil. The prokaryotic cells, your bacteria, you always look at those with oil under the microscope. And they still look really small. The metabolic requirements limit the size. You've got the ratio of plasma membrane surface area to the volume. Smaller cells are going to have a greater ratio. What limits it is how much can cross the membrane. If you don't have enough membrane to get in more nutrients, because nutrients have to be able to cross that membrane, you're not going to be able to get very big. So when you look and you see bigger organisms, it doesn't mean bigger cells, it means more cells in there. A cell can only get so big before its size cannot be sustained by the membrane that can let things in and out. The membrane regulates so much on the cell. The membrane's probably a very close second in importance to the DNA in the cell in terms of how it regulates it and keeps it alive. So eukaryotic cells are going to have more membranes. It's going to have membranes on the inside that are going to separate things into organelles. The membranes are also going to facilitate and participate in metabolic reactions. So you've got membranes in the mitochondria in animal cells, which is where you're going to derive your energy from. So in your eukaryotic cells, your nucleus is going to contain most of the genes. You do have a little bit of DNA outside of the nucleus. You have some in your mitochondria. Your nucleus is surrounded by a nuclear envelope. It's a double membrane of phospholipid, just like the plasma membrane is. And it's perforated by pores, and it's got little proteins that are going to cross that membrane. The way we usually draw that membrane is going to be, we've got these phospholipids, and they're in a bilayer. So you've got the two layers turned towards each other. And then you'll have these membranes embedded in there. And then you also have these pores, which is kind of a weird way to draw it, but maybe that's a little more accurate looking, where things can get in and out of the membrane. So one of the things that's going to have to get through there is your RNA in order for you to be able to make proteins. So the pore complex has an inner and outer membrane that's continuous at the lip of the pore, and you've got intricate pore complex that's going to line it. So it actually is made up of membrane that kind of creates like a little lip on there for going in and out of. And they're not just a free hole for things to go in and out of. It's going to have specific requirements of what goes in and out in there. Your nuclear lamina is going to be a net-like protein filament that's going to be underneath the lining of the nuclear envelope that helps to maintain the shape. If you were to look underneath your skin, you've got kind of a mesh-like areolar tissue underneath there that helps keep your blood vessels in place and keeps things from moving around. You're also going to have kind of a mesh-like underneath here that's going to help hold your nuclear membrane in place and keep it in its shape. And then you're also going to have those that will extend all the way through the interior that make up your nuclear matrix in there. The chromosomes are going to be the organized form of DNA. Most of the time it's going to be kind of loosely coiled all over the place, but when you go to reproduce the cell is when you're going to want to form the chromosomes in there. And there is a characteristic number of those in eukaryotic species. So we all have the same number of chromosomes in there. It's kind of interesting when you look at chromosomes. More chromosomes does not necessarily mean more advanced. So your dogs all have, if you have a dog, it's got more chromosomes than you do. There's just variation in there, and there's no correlation with how they've divided up into their complexity. There's a fern species that has well over 1 million chromosomes in there. And then a lot of bacteria just have a single chromosome. The chromatin is the complex of proteins in the DNAs. It's the loose, diffuse mass of DNA in there. The way I like to think of it is the chromatin is kind of like when you have all your books and your materials out and you're studying, you can actually go in and access the things. However, when you put them all away and you put them all into binders and folders and all nice and neat, it's nice for moving them or storing them or putting them somewhere else, but you can't actually access the information in there. And it's the same way when you have them as chromosomes. Your cells can't actually get in and read the DNA at that point. It's just interested in transporting it and dividing it. When it's chromatin, you can actually get the enzymes in there to physically reach the DNA. Your nucleolus, or if it's multiple, we call it nucleoli, is seen in the non-dividing cells. It will go away when a cell starts to divide. It's going to synthesize your rRNA, which is ribosomal RNA. It's going to help to import proteins into there to assemble the large and small subunits of the ribosomes. And then they're going to exit through the nuclear pores out to the cytoplasm and assemble into full-size ribosomes. The nucleolus also is going to help to regulate cell division. So your mRNA, your messenger RNA, is going to be synthesized in the nucleus, and it will get transported out to the cytoplasm for the ribosomes to translate. So your DNA in there has the instructions for making your proteins. However, you can't set that out through this pore. You're going to make an mRNA to send the instructions out to the ribosomes who make your proteins in there. So those ribosomes are little protein factories. Did you need to get to the microscopes, Linda? Oh, it's okay if you look at them in here. No, it's okay. I do open lab times when I have class. If you're waiting for it to be empty, this room's almost never empty.
No, I should have said open lab.
Yeah.
Okay, come on, put your side.
So with the ribosomes, they're the little protein factories. Some of them are going to be attached to membranes on there, and some of them are going to be freely bound, floating around. So the free ribosomes are going to be suspended in the cytosol, and they're usually going to catalyze the first steps in sugar breakdown in there. The bound ribosomes are going to be attached to your endoplasmic reticulum, which we abbreviate as ER. And so when things are going to be inserted into membranes, it will do packaging with the organelles and help to export secretions. So it kind of gives it a membrane pathway to follow in there. So this endomembrane system is the membranes inside the cell. You have different membranes carrying out various tasks in there. Protein synthesis in there, transporting things, metabolism, moving lipids, detoxification in there. That's one of the reasons why having more surface area is so important to a cell, is these membranes like that. It's going to include various membranes that are not identical in structure. So membranes don't all have to be identical in structure. They're all going to have phospholipid, but the things that stick through here are going to vary in the membranes. Vesicles are just membrane-bound sacs. So sometimes the cell wants to make things that it keeps isolated from the rest of the cell. If you're going to make a poison to kill other cells, you don't want it to kill you, so you put it in a sac. And that's what the vesicles are going to be good for. So your endoplasmic reticulum is the extensive network. It's about half, or a little more than half, of the total membrane in most eukaryotes in there. And what it looks like is you've just grabbed this long bunch of membrane and let it wind back and forth and shoved it inside the cell. The what? Kind of, except the colon only goes around. It's more like the intestines inside of it. And it's actually continuous with the nuclear envelope. So if you've got your cell here and you've got your nuclear envelope, this actually continues out to make this endomembrane. Where it's all phospholipid bilayer in there. Yeah, why is it not that way? You get the point. It's bilayer all the way through. So the endoplasmic reticulum, like that, is actually a hollow tube that you can squeeze things through and have it travel places in the cell. So the cisternal space is that internal hollow space in the middle of it, and it's going to be separated from the cytosol by that endoplasmic reticulum. So this, everything outside of this is separated from what's inside of there. So your smooth ER is going to lack surface ribosomes. The enzymes in the smooth ER are important for lipid synthesis. So ribosomes, you always want to associate those with protein. When you look at what your rough ER is going to be involved with, it's going to have more to do with proteins, where the smooth ER, more of your fats, because there isn't the ribosomes on there for proteins. It's going to be involved with steroids and phospholipids in there. When you detoxify drugs in the liver, it's going to attach an alcohol group to it. It increases the solubility and facilitates the flushing. So one way to be able to help make something water soluble so that you can flush it out of your body is to put a hydroxyl group on there. It's more likely to be water soluble. So when you take in a toxin, that's a way for your body to help be able to flush it out in there. Proliferation of the smooth ER can increase tolerance to certain drugs. So that's one of the reasons why with certain drugs you need to have higher and higher amounts of it, is it's involved in processing that drug. When you take a drug, you are taking in a toxic substance for a desired effect in there. So you could OD on virtually any drug. You want that toxic effect to modify something in your body. And your body tries to handle that toxin, so as your liver gets more efficient at dealing with it, it's going to make more smooth ER in there, and then it will also need higher amounts of that drug to achieve the same effect in there. It's going to store calcium ions that are going to be specialized in muscle cells. We actually call it the sarcoplasmic reticulum in muscle cells, and it stores the calcium you need to be able to contract. So the rough ER is going to have those ribosomes on the surface. So it's going to be more involved with the protein secretions in there. It's going to help to make insulin, your glycoproteins, things that are in your proteins and carbohydrates in there. It's going to make the secretory proteins, and it's going to keep them separated from the cytosol. So when you make them, you can also use some of this membrane in there to wrap them up and keep them isolated in there. It will also help with membrane synthesis to add more proteins and phospholipids onto its own membrane in there. So a cell can have both rough and smooth ER. It's not going to have exclusively one or exclusively the other. You'll have a mix in there, and they just have different functions. And then you usually have some free ribosomes that will have their own separate functions in the cell too. So the Golgi apparatus, this looks like a bunch of flattened out pancakes, kind of a stack of pancakes in the cell when you look at an electron microscope image of it. The flattened membrane sacs are referred to as cisternae in there. They're going to help to modify and store proteins and send them to other destinations. So when you make a protein in there, this is going to help to finish the job and maybe stick a couple different proteins together in there, giving it that quaternary structure. It will also help guide it where to go in the cell. So these are going to be extensive and secretory cells, because if you're making a lot of secretions, you need to be able to get those secretions ready to go somewhere else. They have a definite polarity, differing in composition. So they have a cis and a trans face. It isn't that things pass back and forth through it randomly. It goes in one direction in there. So the cis is going to receive vesicles, trans gives rise to vesicles. It's going to come in the cis, out the trans end. It helps to modify the carbohydrates in your glycoproteins. It will help to alter your membrane phospholipids. It's going to manufacture certain macromolecules and secrete many polysaccharides. So it's kind of like your little finishing center. When your proteins are made with the ribosomes, they're not always polished and ready to go. The Golgi apparatus can take and finish stuff up and say, okay, now it's gone through its quality control, it's ready to go out into my world. So the cisternal maturation model looks at the cisternae of the Golgi as it progresses forward from the cis to trans phase, modifying the cargo as they go, and it marks products with molecular ID tags to direct them where to go. So it kind of puts the label on it so that when it exits your Golgi, it's not floating around randomly. It's got the instructions of where to send that protein. So the lysosomes, these are little membrane-bound sacs of hydrolytic enzymes. And what they're going to do is digest macromolecules in animal cells. So lyse means to split. So a lot of times by digesting these macromolecules, you're able to lyse or split open a cell, which will kill it in there. So what keeps it from destroying its own membrane? That's the sac around it. There's proteins on the inner surface that help to protect that lysosomal membrane. They're made up of a special type of membrane that's going to be immune to the enzymes they have on the inside. One of the things they'll do is phagocytosis. So food vacuoles are going to form, they fuse with the lysosome, and then the enzymes get dumped in there to digest it. And it will help to break things down into monomers for the cell. So if you've got your cell here, and you've got a chunk of food here, what's going to happen is that lysosome is going to need to come fuse with this. So what it will do is when they get together then, the membranes fuse, and it allows the contents to get dumped in. So that way you can digest what's in here, but not digest everything else in your cell at the same time. Sometimes we do want to digest what's in our own cell. So if one of your cells has turned bad, you want to be able to eliminate it in there. And autolysis or autophagy is when the cell uses the hydrolytic enzymes in there to recycle its own materials. And this is actually really important that this happens. One example of a disease from this not happening is Tay-Sachs disease. It's got a lipid digesting enzyme missing in there, so you don't actually break those down and the lipids start to accumulate in the brain for the person. Autoimmune diseases can be the result of failure for that to happen. So if one of your immune cells recognizes your own tissue, you need to do autophagy on there or autolysis and destroy that cell, otherwise your body is going to continue to attack itself in there. So it can be really important for those things. A lot of times cancer cells will be triggered to go into this early on as a way that your body helps to eliminate those. They figure most of us probably have some cancer cells floating around in us at any given time, and this is how we keep from ending up with tumors all the time. It's one of the ways that the body protects itself from it. It's also important with like embryological development. So some of the cells, if you look at an embryo, you'll notice they have webbed fingers and webbed toes in there. The reason we don't all come out with webbed fingers and toes is that process goes on to erode this tissue in between so that you have normal fingers. So vacuoles, where you're going to see a lot of these is food vacuoles. They talk about them mostly in plants. They're formed by phagocytosis of food, and that's what also gives us some of the nutrients when we eat the plants, is their vacuoles where they store these sugars in there. We have contractile vacuoles that are going to pump excess water out of the cells. This is something you'll see in freshwater produce, little single-celled organisms that live in fresh water. They need to pump the water out so that they don't take in too much water and burst in there. The central vacuole in plants, this is going to be a coalescence of smaller vacuoles that carry out hydrolysis. It's going to hold ions in there, help to dispose metabolic byproducts. It will contain pigments in some cases, poisonous compounds in some cases. They're basically just big packages. So the mitochondria, these are probably the most interesting organelle. I like them best because it's where you make energy in there. More mitochondria you have, the more energy you can make. Mitochondria are your friends. We like them. So it's the site of cellular respiration, which is the type of metabolism that you do. More mitochondria are going to be formed in tissues that do more cellular respiration. In plants, they have chloroplasts, which is where they do their photosynthesis. So they get two different structures for plants and animals in there. They're going to be enclosed by a membrane, but they are not considered part of the endomembrane system. Their membranes are going to be made by free ribosomes. They contain a small amount of DNA. Yeah. So when you get your mitochondria, you've got sperm cells and egg cells that make up you. Basically, the sperm comes along and donates the genetic information, but the egg has everything else. So the majority of your mitochondrial DNA you actually get from your mother. So if you're looking in your cells for similarities, we actually carry more of that similarity from our mother in there. When they look at a lot of the stuff with evolution, they're looking at the mitochondrial DNA because you can see that being directly passed down the line. So I think that means men should be changing their names when they get married. I'm kidding. Most men don't find that to be very funny. So these organelles, they contain a small amount of DNA, and they're semi-autonomous. They grow and reproduce within the cell. So if you need more mitochondria, you can grow and reproduce more mitochondria within your cell. You don't have to make a new cell to do that. So when you exercise, one of the things that your body is doing to adapt to it is making more mitochondria. Eating them doesn't get you more mitochondria. You have to induce your body to make its own. And then we have peroxisomes that are another oxidative organelle that's not part of the endomembrane system. These are going to make hydrogen peroxide in there as a way for the cell to help protect itself. So your mitochondria, to me, for some reason, when I look at these, I think sausage with mustard. They've got this internal membrane in there with them. So it's enclosed by two membranes. You actually have a phospholipid bilayer on the outside. There. It's a lot more uniform in size than my drawing is. And then you've got this membrane on the inside as well. And it's going to form these smooth inner folds, and we call these folds the cristae in there. So your intermembrane space is the region between the inner and outer membrane. So this is your intermembrane space out here. And then the mitochondrial matrix is enclosed by the inner matrix. It's going to have some enzymes, some DNA, and it's tidally folded for a large surface area. So this is where you're going to have the matrix in the middle of it. So the chloroplasts for photosynthesis, they're a member of the plastids organelles. You've got amyloplasts that store starch. So your starchy plants that you would eat, like potatoes, are going to have more amyloplasts in them. The chloroplasts are going to store pigment. So when you look at the same plant, down in the bottom in the root in the ground, you're going to have the amyloplasts. There's no point in having chloroplasts there. It's not getting any light. The part of the potato that's above the ground is going to have the chloroplasts that are going to actually use the light for making the energy in there. So the thylakoids are going to be the interconnected membrane sacs inside. And the granum are the stacks of thylakoids, or grana. So when you look at those, they just kind of look like you've got, and they're interconnected, they're not entirely separated in there. So they'll just be these stacks of the pigments like that in there. The stroma is going to be the fluid outside. So when it's surrounded by its membrane out there, you've got stroma out here, you've got your thylakoids, the granum is the stack in there. And it's going to have your DNA, ribosomes, and enzymes in there. So we're going to go more into what goes on in the mitochondria and the chloroplasts with metabolism. So the peroxisome, it's a specialized metabolic component. It's bound by a membrane in there that produces hydrogen peroxide. So what would you use hydrogen peroxide for? So you use it to try and clean things because you're hoping it's going to kill bacteria. It kills some bacteria can metabolize it in there. You use it to oxidize things, and that's how it kills them. So it will use some of the oxygen to help break down fatty acids in there. It's not going to metabolize them and burn them up. It just helps to split them so that your metabolism can deal with them. It also helps in detoxifying alcohol in the liver in there. And then they are part of the immune protection in there with the bacteria. Glyoxysomes, these are specialized in the fat storing tissues of plant seeds. It helps convert the fat to sugar to feed the seeds. So one of the reasons why when you eat seeds they have so many nutrients is the seed is kind of like the ovary, or not really the ovary, but it's almost more like the uterus. The fruit's the uterus, and then it's got the seeds in there, where you've got the baby. It's got all the stuff to feed that baby plant in there. And that's why you have those glyoxysomes in there to help break down the nutrients to feed that baby plant. So you have a cytoskeleton. This is going to be a fiber network inside the cytoplasm that's going to help to organize things. It's like a little mini skeletal system inside your cells. It's much more developed in eukaryotic cells than it is in prokaryotic cells. So it's going to help provide mechanical support for shape. So some cells are going to be round and look like a blob, but some are going to have a more defined shape, and this helps to give it that. Helps with motility by interacting with the motor proteins, so making your cilia and flagella. It's going to provide transport pathways inside the cell. It's going to move plasma to do phagocytosis. And it helps to regulate biochemical activities in response to mechanical stimulation. So there are some cells, you can't necessarily see it in a human real easily, but there's certain plants when you poke them, they automatically move in there. There's some plants that when you poke them, they'll shoot out little things at you. A lot of cactuses have things like that. This is going to help with those mechanical activities in there when you irritate a cell to have it respond back. So the components of it, you've got microtubules. These are going to be the thickest fiber. They're made of tubulin protein. They're going to help provide shape and support, tracks for the organelles to move. So the organelles aren't just floating around and ending up in random places. They're actually traveling on a pathway in the cell. It's going to help to guide secretory vesicles so that when you want a secretion to leave the cell, you put it on a track. It's kind of like a conveyor belt that's going to take things away in there. You've got the centrosome in animal cells. It's a region that microtubules are going to grow out of. And within the centrosome, you have the centrioles that are going to be composed of 9 sets of triplet microtubules in a ring. So what you've got are like these little triplets in a ring. So they're really small. I know that it's hard to see that, but it's just little pairs of 3 or little sets of 3 in there, and there's 9 of them that make up a ring like that. So these are going to be involved with animal cells in the division and replication. They're not essential for it because we know that yeast cells and plant cells don't have them, but they are important for animal cell replication. And we'll look at that in a lot more detail later on too. So your cilia and flagella, these are going to be microtubule containing extensions that project from some cells to propel them. Cilia will tend to be more of a hair-like structure all the way around it. Flagella are going to be more of a whip-like tail. Your classic example of flagella is sperm, and it moves in an undulating motion for swimming in there. It's moving the same direction as its axis. So when you look at the sperm, the axis is this way. The sperm should go that way. Where with cilia, it works more like oars. The axis is this way of them, but it will propel it that way. And they're going to have alternating recovery and power strokes in there. So your primary cilia are going to act as signal receiving antenna and transmit molecular signals that appear crucial in brain function and embryological development. So some of them aren't going to actually move things. It's just that they're going to be little receptors on there that when you bump them, it tells them something's next to it. So in embryological development, that's going to be important because it's going to help the cells know who are their neighbors around it and when to change modes in there. So these microtubules are arranged in what we call a 9+2 pattern in there. So you have your 2 in the center, and then 9 pairs around the outside. And you're going to find that in nearly all eukaryotic flagella and motile cilia. The 9+0, you've just got the 9 pairs around the outside. And they have nothing in the middle. So that's going to be more in your non-motile primary cilia in there. So when you look at these in your membrane, so we're going to say this is your membrane, you're actually going to have this basal body that it attaches to in there, where you're going to have one of these hooked to that basal body. It's going to anchor that assembly onto the cell. And then you've got these little dienins that are protruding motor proteins along the length. They're going to help give that bending movement in there that are going to cause it to bend. And it's going to change shape when you use ATP for energy. So when it's got energy put in it, it's going to be one shape. When the energy is used up, it goes to the other shape. And it goes back and forth like this to cause that waving motion in there. So a little bit smaller, we've got microfilaments that are solid rods made up of actin protein. It's a globular protein that's going to be in twisted chains. So it's going to be more kind of like twisted little chains like that you're going to see this one. It's the same actin that's used in muscles. These are going to help to support cell shape. They're going to give the cortex, the outer cytoplasmic layer, a gel consistency in there. They're going to bear tension forces. So if you're going to be a cell that's going to engulf something and you're going to wrap part of yourself around it, you want to have some of these in there so that when you're wrapping around something, you don't just lob off part of yourself. They're going to be in the core of the microvilli to increase surface areas. So you've got some cells in your digestive system, your small intestine, that are going to have these little finger-like microvilli on there. You're going to have these in there so that these don't just break off, because otherwise they would be really delicate, and when you'd eat your food would just scrape them off and they'd be gone. They're going to be involved with motility. They're part of the contractile apparatus of muscle with actin, so they do kind of pulling motions in there. And then they'll do amoeboid movements by extending the pseudopods in there. So that's that wrapping around thing that they'll do. And then they also do cytoplasmic streaming. So your cytoplasm inside your cell, this is how it gets circulated around. And then your intermediate filaments, they're a little bit larger than the microfilaments in there. They're also going to help to bear tension, keep the cells from tearing too easily. These will persist after the cell dies, so it's kind of like your skeleton. It persists after you die in there. It makes up the nuclear lamina. So where we talked about your nuclear membrane having a lamina underneath that helped make it stronger, that's what this is going to be made of, and it's going to help with the shape and framework. And that's a good place to stop for today in there. So those are things within the cell.